Microbiology for GNM Unit 5, Control and Destruction of Microbes. Principles and methods of microbial control. Microbial control refers to the techniques used to eliminate, inhibit, or reduce the number of microorganisms to prevent infections, contamination, and spoilage. The key methods include sterilization, disinfection, chemotherapy, and pasteurization. Sterilization. Sterilization is the process of completely killing or removing all microorganisms, including bacteria, viruses, fungi, and spores. Methods of sterilization. Physical methods. Heat sterilization, moist heat, more effective than dry heat. Autoclaving, 121 degrees Celsius, 15 pounds, 15 to 20 minutes, kills all microbes and spores. Used for surgical instruments and culture media. Boiling, 100 degrees Celsius, for 10 to 15 minutes. Kills most vegetative bacteria, but does not kill spores. Tyndallization, intermittent boiling at 100 degrees Celsius for 30 minutes for 3 consecutive days. Used for heat-sensitive materials. Dry heat, hot air oven, 160 to 170 degrees Celsius for 2 to 3 hours. Used for glassware and powders. Incineration, used for complete destruction of contaminated materials, e.g. needles, bandages. Filtration membrane filters, 0.22 micrometers, 0.45 micrometers pore size. Used for sterilizing heat-sensitive liquids and air. Radiation ionizing radiation, gamma rays, X-rays. Used for sterilizing disposable medical equipment. Non-ionizing radiation, UV light at 260 nanometers. Used for disinfecting air, water, and surfaces. Disinfection. Disinfection is the process of eliminating or reducing most pathogenic microorganisms, except bacterial spores, on non-living surfaces. Common disinfectants and their uses. Disinfectants, phenol and phenolics, disrupt cell membranes, used in hospitals and laboratories. Alcohols, ethanol and isopropanol, denature proteins, used as hand sanitizers and skin antiseptics. Halogens, chlorine, iodine, bromine, work as oxidizing agents, used for water treatment. And as antiseptics, hydrogen peroxide produces free radicals, used for wound cleaning and surface sterilization. Glutaraldehyde, 2%, acts as a high-level disinfectant and sterilant, used for endoscopes and plastic equipment. Ethylene oxide, ETO, gas, sterilizes heat-sensitive and moisture-sensitive items, used for catheters, IV sets, and gloves. Formaldehyde, formalin, used for surface and room fumigation, commonly used in operation theatres and hospital rooms. Chemotherapy. Chemotherapy involves the use of antimicrobial agents to kill or inhibit the growth of microbes inside a living organism. Types of antimicrobial agents. Antibiotics kill or inhibit bacterial growth, e.g. penicillin, tetracycline, ciprofloxacin. Antivirals inhibit viral replication, e.g. acyclovir for herpes, remdesivir for COVID-19. Antifungals used to treat fungal infections, e.g. amphotericin B, fluconazole. Antiparasitic drugs target parasites, e.g. chloroquine for malaria, metronidazole for amoebiasis. Mode of action of antibiotics: inhibit cell wall synthesis, penicillins, cephalosporins. Inhibit protein synthesis, tetracyclines, macrolides. Inhibit DNA-RNA synthesis, fluoroquinolones, rifampin. Inhibit metabolic pathways, sulfonamides. Pasteurization. Pasteurization is a mild heat treatment process that reduces the number of pathogenic microbes in food and beverages without significantly affecting their quality. Uses of pasteurization prevents foodborne diseases, e.g. tuberculosis, typhoid from milk. Increases the shelf life of perishable foods. Types of pasteurization: low temperature, long time LTLT, bulk method, 63 degrees Celsius for 30 minutes, used for milk and fruit juices. High temperature short time HTST, 72 degrees Celsius for 15 seconds, used for milk and beer. Ultra high temperature UHT, 135 degrees Celsius for 2 to 5 seconds, used for long shelf life dairy products. Comparison of sterilization, disinfection and pasteurization. Sterilization, complete destruction of all microbes and spores, kills all life forms. Used for surgical instruments and culture media disinfection. Reduces most pathogens, but not spores, kills most bacteria and viruses. Used for hospital surfaces and drinking water, pasteurization. Reduces microbial load in food and beverages, kills pathogens, but not all microbes. Used for milk, juices and canned food. Medical and surgical asepsis, Asepsis. Asepsis is the absence of disease-causing microorganisms, pathogens. It refers to practices that prevent infection and maintain cleanliness in medical settings. Types of asepsis. Medical asepsis, clean technique, used to reduce and prevent the spread of microorganisms. Maintain personal hygiene, hand washing before and after patient care. Disinfection of surfaces and equipment, use of PPE, gloves, masks, gowns, proper waste disposal, biomedical waste management, respiratory hygiene, cough etiquette, cleaning from cleaner to dirtier areas. Used in administering medications, providing personal care, feeding and handling linens, minor dressing of wounds, in outpatient, departments and wards. Surgical asepsis, sterile technique, used to eliminate all microorganisms, including spores, from an area or object. Sterile objects must remain sterile, no contact with non-sterile items. Use of sterilized instruments and dressings, donning sterile gloves, gowns and masks, maintaining a sterile field, avoid reaching over a sterile area, use of antiseptics for skin preparation, sterilization by autoclaving, dry heat or chemical methods. Used in operation theatres, invasive procedures, catheterization, IV insertion, wound suturing, central line dressing, delivery rooms, ICU settings. Biosafety and waste management, biosafety. Biosafety refers to precautionary measures taken to prevent exposure to harmful biological agents, such as bacteria, viruses and toxins in healthcare and laboratory settings. Principles of biosafety. Minimize exposure to biological hazards, use PPE and follow proper safety protocols. Containment of infectious materials, use biosafety cabinets and isolation techniques. Proper disposal of biohazardous waste, segregate, disinfect and dispose of safely. Regular training and education. Healthcare workers must be trained in biosafety protocols. Emergency preparedness, plans for accidental spills, needle stick injuries and exposure. Biosafety levels, BSL, BSL-1, low risk, non-pathogenic organisms, e.g. E. coli K12, BSL-2, moderate risk, pathogenic organisms that pose moderate risk, e.g. Staphylococcus aureus, hepatitis B. BSL-3, high risk, airborne pathogens causing severe diseases, e.g. tuberculosis, SARS-CoV-2. BSL-4, extreme risk, highly lethal viruses with no cure, e.g. Ebola, Marburg virus. Biosafety practices. use of personal protective equipment, PPE, gloves, masks, gowns, eye protection, safe handling of needles and sharps, use safety-engineered devices, avoid recapping, proper decontamination and disinfection, use chemicals like chlorine, formaldehyde and alcohol, use of biosafety cabinets, BSCS, for handling infectious agents, vaccination of healthcare workers against hepatitis B, tuberculosis, etc. Waste management. Biomedical waste management refers to the proper handling, segregation, treatment, and disposal of medical waste to prevent infections and environmental hazards. Types of biomedical waste. Infectious waste includes contaminated dressings and blood-soaked bandages, treated by autoclaving or incineration. Pathological waste includes human tissues, organs, blood and body fluids, treated by incineration or deep burial sharps. Waste includes needles, scalpels and broken glass, stored in puncture-proof containers, and incinerated pharmaceutical waste. Includes expired medicines and antibiotics disposed of by incineration or chemical methods. Chemical waste includes disinfectants and laboratory reagents treated by chemical neutralization. Radioactive waste includes radioactive materials from cancer treatment, requires radioactive decay or special disposal. Biomedical waste segregation, color coding system. Yellow bag, for infectious and pathological waste, uses non-chlorinated plastic bag, disposal by incineration or deep burial. Red bag, for contaminated waste like tubings, gloves and IV sets, uses plastic bag or container. Disposal by autoclaving or shredding blue container. For glassware and metallic body implants, uses puncture-proof container. Disposal by autoclaving or recycling, white or translucent container. For sharps like needles and blades, uses puncture-proof container. Disposal by incineration or shredding, black bin. For non-hazardous waste like paper and food, uses general waste bin. Disposal through municipal systems. Methods of waste treatment and disposal. Incineration burns hazardous waste at high temperatures, 850 to 1100 degrees Celsius. Autoclaving uses steam under pressure to sterilize infectious waste. Chemical disinfection uses chlorine, bleach or formaldehyde for liquid waste. Microwave treatment uses heat to disinfect medical waste. Deep burial used for anatomical and microbiological waste in rural areas. Importance of biosafety and waste management prevents healthcare-associated infections, HAIs, reduces environmental pollution and contamination, ensures safety of healthcare workers and the public, complies with national and international health regulations,